Good morning, everyone. It's Monday, June 21st, beginning of our second week of summer school. And due this coming Sunday, June 27th, are your two labs, Lab 3 and Lab 4, glucose and diabetes, and the mystery of the 7 deaths. So please get going on those. And then in the lecture, we're going to continue talking about transport. If we look at transport take 2, it's talking about gated channels. So these are channels that are not always open. They have to be stimulated to be open, whether that's by a ligand, otherwise known as a signaling molecule. It could be a change in voltage, or it could be some mechanical force within the body, like a stretch or pressure that opens up a gate. Exocytosis and insulin release. Exocytosis is the release of contents within secretory vesicles inside of a cell. And the reason I have insulin release, it's very important in our body, but two, it covers a wide breadth of things we've already talked about. It's going to cover leakage channels, it's going to cover ligand-gated channels, voltage-gated channels, exocytosis, depolarization event. So please take a look at that. It's a multi-step and fairly in-depth process. And it's going to take more than just one viewing of that video to really get a good grasp of that. But please take a look at that. Another thing we're going to talk about this week is cellular respiration. And the whole purpose of cellular respiration is to produce ATP. And if you look at equations or books, the suggestion is that cellular respiration will produce upwards of 36 ATP, 34 to 38, but let's just say 36. And that is true in the presence of oxygen. So there are 3 phases to cellular respiration: glycolysis, the Krebs cycle, and the electron transport chain. Electron transport chain we can couple with a process known as chemiosmosis and oxidative phosphorylation. But for simplicity, I would just refer to it as the electron transport chain. The Krebs cycle is also known as the citric acid cycle. So we have glycolysis, Krebs cycle, and the electron transport chain. These are 3 separate but intimately related processes within cellular respiration. Glycolysis happens in the presence or absence of oxygen, and it's always going to produce a net 2 ATP. It's going to produce 2 pyruvate molecules as well. Those 2 pyruvate molecules individually will enter the Krebs cycle. And after one cycle from one pyruvate, we get 3 NADH molecules and 1 FADH2 molecule and 2 ATP molecules. But because there's 2 pyruvate, it's going to go through twice. So the Krebs cycle will produce a total of 6 NADH molecules and 2 FADH2 molecules. The reason I have 8 NADH here is because the 6 from the 2 Krebs cycles, and there's 2 NADH produced during glycolysis, which I think I was remiss to say when I was talking about glycolysis a few seconds ago. So from glycolysis, we get 2 NADH molecules, 2 ATP molecules, and 2 pyruvate. From the Krebs cycles, that's 2 of them, we get 6 NADH molecules and 2 FADH2 molecules and 2 ATP molecules. For a total of the 2 Krebs cycles, we get 6 NADH molecules, 2 ATP molecules, and 2 FADH2 molecules. Combined glycolysis and Krebs gives us 4 ATP, 8 NADH, and 2 FADH2. And I will argue all day long, these 8 NADH and 2 FADH2 molecules are much more important than those 4 ATP, because those 8 NADH and 2 FADH2 are going to result in making about 34 more ATP via the electron transport chain and chemiosmosis. Okay, that's it for today. I will not be at my office hours today, and I apologize for that, but we'll resume with office hours tomorrow on Tuesday, June 22nd.